So, hello students, welcome back to your own channel Vedantu NEET English. This is Baswaratso, your biology master teacher. So, quickly let me know in the chat right now if my voice is audible to all of you and the presentation behind me is visible to each and every one of you. Now students, the most, well, what do I say, not most difficult, it's a chapter which is continuation to your previous chapter, that is morphology of flowering plants. Now in morphology of flowering plants, we learnt about how the plant looks from the outside. Now this is the chapter which is the anatomy of flowering plants, where we look how the plant looks from the inside now. Now let me give you a quick perspective. Perspective how the variation in plant takes place. Now for example students, once again, let me just check if we are live. Let me just check. Yes, we are live. Beautiful, beautiful. Now students, let me give you a small perspective. For example, you're taking a walk in a park. And you look at a plant. You look at a beautiful plant. Now you look at the shoot system, you look at the root system. In the root system, you find roots. In the shoot system of a plant, you find the branches, you find the stem, you find the leaf, you find the flower. That is in the morphology. Now I'll ask you one question, students. Now I'll ask you one simple question. Now, when you look at a plant, from the morphology outside, can you tell me the number of cells inside? Can you tell me which type of cell is present in the leaf? What is the shape of parenchyma cells? Can you tell me how are the vascular bundles arranged inside the cell? Is it radial arrangement, or is it going to be your conjoint arrangement or collateral arrangement? Can you tell all that? Unless you take the leaf or stem or root and unless you cross section it, you cannot tell how a plant looks from the inside. Yes. And as soon as you cut a plant and you study the internal structure of a plant, when you cut a plant and you study the internal structure of a plant, that becomes anatomy of flowering plants. So in a simple definition, what is anatomy, students? Anatomy is a branch of science. Anatomy is a branch of science that deals with internal structure of a plant. It deals with the internal structure of a plant. Now here, which plants are we talking about? The here plants which we are talking about is mostly the angiosperms. We are talking about angiosperms. Because these are the flowering plants, angiosperms. So this chapter, that is anatomy of flowering plants, mostly deals with the internal structure of the plants. So let me know in the chat how excited are you for today's class, because this chapter is a very easy chapter. It's a very easy chapter. It is only a matter of perspective and how you're approaching the chapter. Right? So let me know in the chat how excited you are for today's class, because it's going to be one of the interesting classes out there when it comes to anatomy. Okay. Now morphology is what? Morphology is the external form and feature, that is morphology. What is anatomy? Anatomy is the internal structure of the arrangement of cells and tissues inside a plant. Now students, when I was teaching you about morphology, did I teach you something called as modifications? Did we learn something called as modifications? Yes, we learnt about root modification. We learned about root modification, stem modification, leaf modification. Under root modification, we learned about for storage as well as support as well as respiration. Under stem modification, we learned about underground stem, aerial stem, subaerial stem. Under leaf modification, we learned leaf for storage, leaf for protection, right? A leaf which can do photosynthesis, right? So we learned about modifications. Now, listen to me. If a plant is modifying from the outside, do you think the anatomy also will get modified? Obviously. It cannot be said that, just because only morphology can change, anatomy will still remain the same. It is not possible. So if the morphology of a plant is changing because of adaptation, even the anatomy is going to change at the same time. Okay. So apart from normal plants, a normal plant will have three basic things: epidermis, cortex, and stele. A simple plant, if you take a cross section of a plant, it will have epidermis, the outer layer, cortex, the middle layer, and stele, the inner layer. Three different layers of a plant section. It could be stem or root or leaf. Unless there is some adaptation. If there is an adaptation, in that cases, the internal structure might vary. Is that point clear to all of you? Tell me. Is that point clear to all of you? How the internal structure will vary in the case of plants when we compare it with the morphology. When we compare it with the morphology. Now students, how many of you seen this while eating bananas? While eating banana, how many of you seen this? Trust me, tell me in the chat. So when you're trying to eat a banana, usually you encounter this thread-like structures which you remove and throw it out. What are the thread-like structures, students? These thread-like structures are nothing but phloem bundles. That's right. These are the same phloem bundles which are called as complex permanent tissues. Complex permanent tissue, the phloem, whose main function is transportation of nutrients or the food. So these are the phloem bundles. So next time when you're eating banana, don't try to throw it out. You can also try eating it. Okay, you can also try eating it. So that is your phloem bundles. It's nothing but phloem bundles. Right now, students. Now why did I talk about phloem? Because phloem is part of your tissue, which I'm going to teach right now. Now, if I ask you a simple question, students, if I ask you a simple question, what is a tissue? What is a tissue, students? What is a tissue? What is a tissue? If I ask you a simple question, very simple, no complex, the simple of simple question, that is what is a tissue? What is a tissue? Tissue. Tissue is nothing but, can I call it a group of cells? Can I call it a group of cells? Yes. Tissue is nothing but a group of cells. Yes, it is a group of cells. Now here you need to understand there are three things here. Tissue is a group of cell which can have, first thing, common origin. Yes, they will have common origin. For example, the entire shoot system is technically made from shoot apical meristem. Yes, as the shoot apical meristem is growing, it is giving rise to the entire shoot system. They will have a common origin. Yes, they will have common origin. Yes, they will have a somewhat common function. Yes, they will have common function. They will have common function. Now, the third point, will they have common structure? That is very interesting to note that. Do you think they will have all the tissues, do you think they have common structure? Common structure. Do you think they have common structure? Because when you were in your class 10th, class 9th, you will learn that tissues have common origin, they have common function, and they have common structure. But as you're coming to the older classes, you will realize it is not the case. Common structure, it can be present and it cannot be present also. Both. It can be or cannot be. The common structure is present. Let me tell you, the common structure is present in the case of simple tissues. In the case of simple tissues. In the case of simple tissues, the common structure is present. It can be present. It can be present. For example, in the case of parenchyma, in the case of collenchyma, in the case of sclerenchyma, common structure is present. But in the case of complex tissues, the common structure is not present. It is not present. The common structure is not present in the case of complex tissues. Now, what are these complex tissues, students? The complex tissue which I am talking about here is nothing but xylem and phloem. So, students, listen to me very carefully. Tissue is a group of cell which has a common origin, common function, common structure can be present, cannot be present. When is the common structure present? In the case of simple tissue, parenchyma, collenchyma, sclerenchyma. The common structure is not the same in the case of complex tissue. For example, in the case of xylem and phloem, the common structure is not present. The common structure is not present. Is that point clear to all of you? Quickly tell me in the chat. A simple definition of a tissue, how does it originate, the function as well as the structure in the chat. Quickly in the chat. Yes. Now, depending upon this, let's try to classify the tissues now. Can we? Yes. Is this point clear to all of you? Yes. That's regarding the tissue. Now let's understand something called as types of tissues. Let's understand something called as types of tissues. Types of tissues. Okay. It's given somewhere here. Let's understand something called as types of tissues now. Now this types of tissue classification is mainly based on its ability to divide. Simple. This is we're doing types of tissues now. Types of tissue. The types of tissue which we are telling right now is mainly based on ability to divide. Ability to divide. That is the main base of classification, ability to divide. Okay. For example, the entire tissue system can be divided into two types. Entire tissue can be divided into two types. We have something called as meristematic tissue. Something called as meristematic tissues. We also have something called as permanent tissue. Permanent tissues. Meristematic tissue as well as permanent tissue. Now what is the difference between a simple meristematic tissue and permanent tissue? Let's understand. Let me draw a box here so the confusion is not there. Okay. What is a meristematic tissue and what is a simple tissue? Okay. Now students, remember, meristematic tissues are basically the dividing cells. They are the dividing cells. These cells are living cells. They are living cells. Now what about in the case of permanent tissue? Permanent tissues have a specific function. They have a very specific function. They are permanent job. They know what to do in life. They have a very fixed job, fixed structure. They also have fixed structure. And your permanent tissues can be living or they can be dead. They can be living or they can be dead tissues. Living or they can be dead tissues. Is this point clear to all of you? Tell me in the chat quickly. Simple, right? This can be they are living cells, they are dead cells, dividing cells. They have fixed function, and structurally and functionally they are done. They are done. Now students, your permanent tissue can be further classified into two categories. Permanent tissues. They can either be simple tissues, or they can be complex tissue. Under simple tissue we have parenchyma. Under simple tissues we have the parenchyma, we have the collenchyma, and we have the sclerenchyma. Now under complex tissue we have the xylem as well as phloem. That is nothing but your permanent tissue. Have fixed function, fixed structure, living or dead tissue. We divide into simple and complex. Simple we have parenchyma, collenchyma, sclerenchyma. Under complex we have xylem as well as phloem. Done. Xylem as well as phloem. Now what about your meristematic tissue, students? Meristematic tissue can also be further classified. Into something called as primary meristem, as well as secondary meristem. Done. Primary meristem as well as secondary meristem when we come to meristematic tissue. Primary meristematic tissue, secondary meristematic tissue. Quickly tell me in the chat, is there any doubt regarding the following two different types of tissues? It's a very, very easy chapter. We'll go very systematically about it, and I believe there will be no doubt regarding any of the following topics when we finish the chapter. Okay, that is the types of tissue based on ability to divide. Now students, we spoke about meristematic tissue. Right? Now Mesenchymal tissue, let's write a few points about it. For example, mesenchymal tissues are dividing. Yes, they are dividing. Intercellular space is almost absent, or it is not present at all, not present. They are very thin cells, dense cytoplasm, large nucleus, vacuoles are completely absent. Now all of these following points are very important, so let's write down one by one. So you also need to write in your books so that you make a good notes out of it, right? You have to make good notes, Madhumita, Divya. Yes, all of you need to make good notes. So make good notes here. Okay. First of all, mesenchymal tissue, I want you to tell me, is it a living tissue or dead tissue? It is a living tissue or a dead tissue? So I told you mesenchymal tissues are dividing cells. They are obviously living tissues. They are obviously living tissues. Living tissues. They are dividing cells. They are dividing cells. They are dividing cells. Yes. Now, they are dividing cells. Now, what about the cell size here? Do you think there will be very large cells or very small cells? Obviously, there will be very small cells. There will be very small cells here. There will be very small cells. I told you intercellular space will be completely absent. Intercellular space will be absent here. Dividing cells. They will have dense cytoplasm. They will have dense cytoplasm. Dense cytoplasm will be there. If there is dense cytoplasm, if there is newly cell formed, do you think they will have thick cell wall or thin cell wall? Obviously, they will have very thin cell wall. Thin cell wall. And if the cell wall is present, usually if it is a dividing cell, the cell is very young. So do you think it will have primary cell wall or secondary cell wall? Obviously, if a cell is very young, it will have primary cell wall. Primary cell wall. Yes, living cells, dividing cells, small cells, intercellular space is absent, dense cytoplasm, thin wall cells, as well as primary cell wall. Now students, what about the nucleus? They'll have large nucleus as well as the vacuoles. Vacuoles is going to be absent. Vacuoles is going to be absent here. They don't require vacuoles right now. They don't require vacuoles. Okay? Done. Vacuoles are absent. These are different properties of your mesenchymal tissue. Different properties of mesenchymal tissue. If you have done writing, I will go to the next slide. Done. Understand every single point related to the meristematic tissue. Related to the meristematic tissue. Done. Now students, see the diagram of mesenchymal tissue. Very thin cell wall here. Do you think they will have very thin cell wall? Very thin cell wall. Yes. They will have very large nucleus. No intercellular space. There is no intercellular space between any of the following cells, and they'll have dense cytoplasm, thin walls, intercellular space is absent, no vacuoles, cells are small and thin walled. All of these properties needs to be in your mind every single time when I ask you about properties of meristematic tissue. Done. Properties of meristematic tissue. Now students, I want you to understand, you can classify the meristematic tissue. You can classify the entire meristematic tissue into two categories. You can divide the meristematic tissues into categories based on two main things. The first type of classification is based on origin. And the second type of classification is based on the location, based on where you find it, based on location and based on origin. Based on origin, we have primary tissue and secondary tissue. Based on location, we have apical meristem, lateral meristem, as well as intercalary meristem. Following. All of this will be very easy. It sounds very fancy and big for you, but once you write down the points and understand each one of them, it becomes very, very easy. Concept will be very clear for you. So quickly write down now. Start writing down. First, we'll do primary here. Write down, start writing down. Now, first based on origin. First, let's start based on origin. Based on origin. Classification of meristematic tissue based on origin first will start. Based on origin, right? Based on origin, they can be broadly classified into primary meristrum, as well as secondary meristrum, Now, you tell me, in your early classes, when you are going to school, do you directly go to secondary or do you go to primary first? Obviously, when you join a school, you will be in nursery, LKG, UKG, then your primary, then secondary class, right? Then secondary you go. So here also in the case of plants life, we have the primary, then we have secondary. So what is primary, students? What is primary? Primary is nothing but it is the meristem which is formed, which is present from the beginning. It is a meristem which is present from the beginning. Now, what about secondary meristem? Secondary meristem is a meristem which is formed later. It is formed later in the plant's life. So first we have the primary meristem which is present from the beginning, then we have secondary meristem formed later in the plant's life. That is a simple difference first. Simple difference between primary and secondary meristem. Now, if primary meristem is present from the beginning, will the plant grow directly, start growing big, or will it first increase in height? First, will the plant start directly growing thick trunks? No. If a plant is very young, starting it will start growing in height first, right? So your primary meristem is responsible for primary growth of the plant. What is primary growth? Primary growth is basically increase in height of the plant, and primary growth is seen both in the case of monocot as well as dicot. In both monocot and dicot, we can see the primary growth. Done. Now, what about secondary growth? What is the main responsibility of secondary growth? Secondary growth is responsible for the lateral growth of the plant. It is responsible for the lateral growth. Now, what is lateral growth? Growth basically in the girth. First, the plant is very thin. Later on, the plant will become very thick. The trunk is forming, right? Thick, woody trunk. That is because of secondary growth. So, this secondary meristem is mainly due to the main function is secondary growth, and it is only found in the case of dicots. Done. Is this point clear to all of you? Primary meristem, secondary meristem, present from the beginning, formed later in the plant's life. Responsible for primary growth, responsible for lateral growth. Found in monocot and dicot, it is found only in the case of dicots. That is secondary growth. Done. Now, this was based on the origin. Now, we can classify. Now, let's classify the meristem. Based on the location. Like, where exactly do you find it? Where exactly do you find the entire meristem? Now, based on the location, it is broadly classified into three categories now. Okay. Broadly classified into three categories. Clear? Classified into three different categories. Now, the first one is called as apical meristem. The second one is called as intercalary meristem. And the intercalary meristem. And the third one is called as lateral meristem. Here intercalary meristem and apical meristem as well as apical meristem. Intercalary meristem. Now let's understand where do you find it? What is the location? Let's find the location of each one of them. Let's find the location of each one of them. Now when you think of apical, when you hear word apical, what comes to your mind? Apex. Apex means tip. Apex means tip. Yes, apex means tip. So apical meristem is generally found in the growing tip and growing tip of the plant. For example, look at the diagram. This is the soil. Yes, we have the soil here. Now here we have the shoot is growing on top. Shoot is growing on top. Yes, shoot is growing on top. Down we have the root. Down we have the entire root system. Down we have the root system. Yes, down we have the root system. Okay. Now students, in the growing regions right here, can you see? In the growing regions of shoot and root, in the growing regions of shoot and root, we find the apical meristem. In the growing shoot region, we find shoot apical meristem, and in the growing root system, we find root apical meristem. So in the growing shoot and root, we find the apical meristem. In the shoot we find shoot apical meristem, and the root we find root apical meristem. Is that point clear to all of you? Tell me. That is the location of your apical meristem. It is found in the apex region or apical region or apex region. Apex. The meaning of apex or apical is tip, front tip. Okay. And your apical meristem is a type of primary meristem. It's a type of primary meristem. That is very important to understand because it is found in the growing shoot and growing root. So it is a type of primary meristem. Now students, what about in the case of intercalary meristem? Intercalary meristem, students. Intercalary meristem is found between permanent tissues, permanent or mature tissues. For example, if this is the entire stem, of the plant, now here we have the nodal regions. If this is a stem, we have nodes here. We have the nodes. Yes, this right here is the nodal region, node, and between the two nodes is the internodal region, internode. So at this nodal region, students, at the nodal region right here, we find the intercalary meristem. If it is present at the nodal regions, as the intercalary meristem is growing, can it increase the length of internode? Yes, it can increase the length of internode then. So imagine in the nodal regions we have the intercalary meristem. If intercalary meristem is growing, will it increase the length of internode? Yes, it will increase the length of internode. It will increase the length of internode. Increase the length of internode. Done. Now one more important point which I want you to understand here is this intercalary meristem. This intercalary meristem is also a type of primary meristem. It is also type of primary meristem right here. Type of primary meristem. Done. It is also type of primary meristem. And this particular type of intercalary meristem, it is mostly found in the case of monocots. Now you see the grasses, right? Grasses and sugarcane. If a cow is coming and it is eating the grass on top, but after a few days you notice the grass is already growing very fast. Why? The reason why the grass is able to grow very fast is because intercalary meristem is there, and because of this intercalary meristem, the internode length in the grass is increasing very fast. Did all of you understand that? Yes or no in the chat quickly? Did you understand the concept behind the intercalary meristem? That is increase in the internodal length in the monocots, especially in the case of monocots. Especially in the case of monocots. Clear? Now, what about lateral meristem? Lateral meristem is very simple. Basically, lateral meristem is present on the sides. Present on the side. For example, students, if this is the stem, this is the stem. Here we have the nodal regions. This is a node, this is a node. Your intercalary meristem will be found here. Intercalary meristem will be found in the sides. It will increase the girth of the plant. It is responsible for the lateral growth of the plant. Okay, responsible for increase in lateral growth. Lateral growth. A good example is going to be vascular cambium. Vascular cambium, which I'll tell you later on. Vascular cambium as well as cork cambium. Both are a type of, both vascular cambium as well as cork cambium are examples of lateral meristem. And remember students, this lateral meristem right here, it's a type of secondary meristem. It's a type of secondary meristem. So what we learnt in love students? We learnt about, based on origin we have primary meristem, secondary meristem, and based on location we have apical meristem, intercalary meristem as well as lateral meristem. Apical meristem is found in the growing shoot and root tip, that is root shoot apical meristem, root apical meristem, and it is a type of primary meristem. Done. Now, what about intercalary meristem? Intercalary meristem is found between the permanent tissue or mature tissue, especially at the nodal regions. And what is the function here? The main function of intercalary meristem is to increase in the length of internode. Increase in the length of internode. And that is why students, when the cow is grazing on the grass, within a few days you can see the grass is growing already. Why? Because of the intercalary meristem. And the last one we have the lateral meristem. While your apical meristem and intercalary meristem both are a type of primary meristem which are present in the young plants responsible for the primary growth, lateral meristem is a type of secondary meristem. Lateral meristem is a type of secondary meristem which is responsible for the growth in the girth of the plant. Girth of the plant. Right? It is present on the side. Can you see it is present on the sides here? Increase in lateral growth. Example is vascular cambium and cork cambium. This cork cambium in the future will make the bark or the phloem, while your vascular cambium will make secondary xylem and secondary phloem. That you will learn later on. Okay. Now remember students, vascular cambium has two types. Vascular cambium has something called as interfascicular cambium and intrafascicular cambium. Both I will teach you today. Okay. Both I will be teaching you today. Done. This is nothing but your meristematic tissues. Meristematic tissue. Now, if I ask you a simple question, can I ask you a simple question now? A simple question I'll ask you now. The simple question is that, do you think the primary meristem gets converted to secondary meristem? Yes or no in the chat quickly? Tell me in the chat quickly. Do you think the primary meristem gets converted to secondary meristem? If you think that, change the thought today only, because there is no way primary meristem can get converted to secondary meristem. It is simply not possible. Why? Because primary meristem can produce primary tissue. A primary meristem will give rise to primary tissue, and that primary tissue, this primary tissue gets converted to secondary meristem. Is the flowchart clear to all of you? We have primary meristem will never give you secondary meristem. A primary meristem will never give you secondary meristem, because a primary meristem will give you primary tissue. Now that primary tissue can undergo dedifferentiation to produce the secondary meristem. remember that point, it will take you long way in botany. Okay, long way in botany. Is that point clear to all of you? Tell me. Is that point clear to all of you? Primary meristem will provide primary tissue. Now that primary tissue will go de-differentiation and we end up getting the secondary tissue, secondary meristem. Okay, secondary meristem. Now I'll give you a quick example for this. Can I give you a quick example? Yes. The quick example is, we have a meristematic cell here. We have a meristematic cell. Meristematic cell. Now this primary meristematic cell, it is a primary meristematic cell. This primary meristematic cell will lose the ability to divide. It will lose ability to divide. Completely lose the ability to divide. And it will end up making something called as permanent tissue. Permanent tissue. Primary permanent tissue. Okay. That is called as differentiation. Differentiation. Then if this primary permanent tissue, if it regains the ability to divide, if it regains the ability to divide, then we get the secondary meristematic tissue. Secondary meristematic tissue. Secondary meristematic tissue. And this is called as what? This is called as de-differentiation. 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 Now if this secondary permanent tissue, if it loses the ability to divide again, that is called as redifferentiation. Redifferentiation. Differentiation, de-differentiation and redifferentiation. The same concept which you learnt in your plant growth regulator chapter. Okay, plant growth regulator chapter. For example, we have any meristematic tissue. Any meristematic tissue. Now this meristematic tissue is dividing and giving you permanent tissue. Permanent tissue will give you example, that is going to be, for example, medullary rays. Medullary rays. Medullary rays are a type of primary permanent tissue. This primary permanent tissue is again regaining the ability to divide. It is regaining the ability to divide. Here, it is undergoing de-differentiation. Medullary rays is undergoing de-differentiation to produce vascular cambium. To produce vascular cambium. Which vascular cambium? Especially it is the interfascicular cambium. Interfascicular cambium. Are you understanding? Interfascicular cambium is obtained like this. Vascular cambium. So this is primary permanent tissue. Now this vascular cambium is a type of secondary tissue. It's a type of secondary meristematic tissue. Secondary meristematic tissue. Did we learn that here? Vascular cambium is a type of secondary meristematic tissue. Also type of lateral meristem. Also type of lateral meristem. That concept needs to be very clear to all of you if you really want to understand the anatomy. If that concepts are not clear, anatomy is going to be a little nightmare. Okay, understand the concept. See here, primary permanent tissue is giving rise to a primary meristematic tissue is giving rise to primary permanent tissue. Meristematic tissue, permanent tissue. Here we have the permanent tissue. This permanent tissue is giving you secondary meristematic tissue. I told you here, permanent tissue is giving rise to secondary meristem. Giving rise to secondary meristem. The process is called as de-differentiation. The process is called as de-differentiation. Done. Increase in the plant girth, lateral meristem, also type of lateral meristem, give rise to secondary tissue. Eventually give rise to secondary tissue. Done? Done. For example, I told you here we got vascular cambium. This vascular cambium will later give you secondary xylem and secondary phloem. This secondary xylem and the secondary phloem are basically a type of secondary permanent tissues. Secondary permanent tissue. And this process from here to here is called as redifferentiation. It's called as redifferentiation. Here to here, de-differentiation. De-differentiation. Now some meristematic tissue to medullary rays, That is differentiation. That is differentiation. Beautiful concept. Now location, you see, at the apical region, at the growing tips, we have the apical meristem. Apical meristem. On the nodal regions, we have intercalary meristem, which is responsible for the increase in internode length. Then we have lateral meristem, which is present on the sides, responsible for the lateral growth. Yes, responsible for the lateral growth. Nice. Now students, let's decode this NCERT diagram now. Let's decode this NCERT diagram completely. Yes, can be. Now I want everyone to focus very carefully now, because this diagram is very important for you to understand. Now here we have something called as, can you see this part here? This part right here is nothing but root apical meristem, RAM. Now this root apical meristem is growing on both sides. It is growing on both the sides. When it is growing on the bottom side, when the root apical meristem is dividing on the bottom side, it produces something called as initials of root cap. It produces something called as initials of root cap. And eventually this initials of root cap will be converted to root cap. Root cap. Done. On the upper side we get initials of central, cylinder and cortex. Then we also have something called as protoderm. Now if you know, the entire root structure has three main parts: epidermis, also called as epiblemma, cortex and stele. Stele is basically vascular bundles. Now here we have the protoderm. Protoderm is nothing but the epidermis. Give rise to epidermis. Then we have cortex, and then we have central cylinder. Central cylinder is nothing but vascular bundles present in the middle. In the middle we have the vascular bundles, then we have the cortex here, and on the outer side we have the epidermis. Epidermis, cortex and stele. That is the root system. On the lower side we have this right here is the root cap. This right here is the root cap. The root cap is developing on the other side. Difficult anything? Difficult? No. First diagram decoded. Now if you look at the second diagram here also we have some growing meristem tissues here. This meristem tissue is nothing but shoot apical meristem, also called as SAM, which is a type of primary meristematic tissue. Okay. Now again it is growing on two sides. On the lower side it is giving rise to differentiating vascular tissues. Vascular tissues are there. Yes. Then we have the upper side. Upper side, I've told you in the previous class, if this is the stem, here we have apical meristem. We have the apical meristem. Now here we have some nodal regions. Yes, nodal regions are there. I have told in morphology, as the shoot apical meristem, as the SAM is growing on top, it is producing leaf in an acropetal succession. I have told you. So as the SAM is growing on top, it is producing leaf in a acropetal succession. Can you see here? That is why on the top we have the leaf primordium. That is, leaves are being arranged. Leaves are being spread out. That is nothing but here, shoot apical meristem. Done. Now students, one more point here I want everyone to focus. As the shoot apical meristem is growing on top, some cells are left behind. Here. As the shoot apical meristem is growing up, some cells are left behind in the leaf axil. In the leaf axis or leaf axil, some cells are left behind. Now the some cells will end up making a structure called as bud. It will end up making the bud here. bud here in the leaf axil, and this bud is nothing but, this bud is called as axillary bud. It is called as axillary bud. And one more thing I want everyone to remember here, this axillary bud right here can end up growing into a branch. It can end up growing into a branch, or it can also grow into a flower. It can grow into a branch, or it can grow into a flower. Branch or flower. Branch or flower. Is that point clear to all of you? Branch or a flower. That is the second diagram. That is the second diagram. Done. Let's read the NCERT lines. Let's read the NCERT lines given here. As you can see here, the growth in plants is largely restricted to the specialized regions of active cell division called as meristems. Meristems is the region of active cell division. Yes. Plants have different kinds of meristem. Meristem which occurs at the tip of root and shoot and produce primary tissue are called as apical meristem. It is called as apical meristem. Apical meristem is the meristem which is present on the growing root and growing shoot. That is apical meristem. Now, root apical meristem occupies the tip of the root, while the shoot apical meristem, that is SAM, distinct most region of the stem axis. That is on the top, shoot apical meristem. During the formation of leaves, along the stem, as the leaves are forming on the top, I told you some cells are left behind. As the leaf is growing on the top, some cells are left behind. Now those some cells are called as what? Those some cells are called as here axillary bud. That is nothing but the axillary bud. Some cells are left behind, and that is called as axillary bud. Called as in the axil of the leaf and are capable of forming a branch or a flower. They are called as axillary bud. Okay. Now the meristem which occupies between the, I told you, the meristem which is present between the two different mature tissues or permanent tissue. The meristem which occurs between mature tissue is known as intercalary meristem. Did we write that? Here. It is found between permanent tissue or mature tissue. Intercalary meristem. Intercalary meristem. Right? Intercalary meristem. It is called as intercalary meristem. They occur in grasses and regenerate the part removed by the grazing herbivores. I told you, if the cow is coming and eating it, if the cow is coming and eating the up part of the grasses, the intercalary meristem will restore by growing, by increasing the internodal length. Both apical meristem and intercalary meristem are primary meristem. Both are the type of primary meristem. Intercalary as well as lateral meristem. Sorry, intercalary and apical meristem. Because they appear in early in the life of the plant. Did we discuss that? Yes. They appear in the early stage of life. That is why they are called as primary meristem and contribute to the formation of primary plant body. What is this primary plant body? Primary plant body is basically increase in the height of the plant. Increase in the height of the plant. Now, meristem that occurs in mature regions, I think is done. Mature regions of root and shoot of many plants, particularly those produce woody axis. Now, when they say woody axis, they are talking about secondary growth here. Secondary growth. Woody axis and appear later than the primary meristem is called as secondary or lateral meristem. Secondary or lateral meristem. Now, here do you want to know a hidden line of NCERT? Do you want to know a hidden line of NCERT right here? Hidden line of NCERT. The hidden line of NCERT here is, here they are telling you, the hidden line of NCERT here is secondary or lateral meristem. When you use, basically when you use or, it says that either of the words can be used, right? Now, with this line right here, would you not think that secondary and lateral meristem are same to same? Do you have that thought in your mind? All of you might be thinking that secondary or lateral meristem are same to same. The meaning is same. To some extent, the answer is no actually. Let me tell you why. Let me tell you why. It tells you that, see this, see the next line. They are cylindrical meristem, okay, fascicular, vascular, cambium, interfascicular cambium, and cork cambium are examples of lateral meristem. Now here it says that interfascicular cambium, fascicular vascular cambium, which is nothing but intrafascicular cambium, and cork cambium as well as cork cambium. They are said to be examples of lateral meristem. Do you not think then all the three of them are technically then secondary meristem then? Do you think that way? Because it says that here fascicular vascular cambium as well as interfascicular cambium and cork cambium, they are examples of lateral meristem. But in the previous line here, they talk about lateral meristem or secondary meristem. Both are present on OD axis. So you end up thinking that both are same to same. But it is not the case. Let me tell you why. Let me write down for you. Here we'll write down. First we'll write down the fascicular vascular cambium. Fascicular vascular cambium. The other name for fascicular vascular cambium is also called as, intra. It is also called as intra fascicular cambium. Both meaning is same. Your intrafascicular cambium and fascicular vascular cambium both are same to same. The same name. Okay, that's the first one. Now second one is called as what? Interfascicular cambium. Interfascicular cambium. And the third one is called as cork cambium. Now technically all three of them are examples of lateral meristem. All three of them are examples of lateral meristem. They're example of lateral meristem. But even though they're examples of lateral meristem, the origin is different here. That's why we classify based on origin and location separately. Why? Because your fascicular vascular cambium, also called as your intrafascicular cambium, is primary meristem, and your inter one is secondary meristem, and this cork cambium is also secondary meristem. It is secondary meristem. Now, if you want to understand better, understand with the help of diagram. This is imagine this is a root section. This is a section right here. It's a section right here. Now here we have the vascular bundles. Here we have the vascular bundles. Here we have the vascular bundles. Right. Here we have the different types of vascular bundles. Okay. Now, here we have xylem, phloem, xylem, phloem, xylem and phloem, xylem, phloem, xylem, phloem, xylem and phloem. Yes, xylem and phloem is there. Yes, we have xylem and phloem. Now, what you want to understand here is there are two types of cambium present here. Students, all of you are able to see this? All of you are able to see this? Yes. Now here I want you to understand which is which exactly. Which is which exactly. If you notice at this green color one, this green color one right here, this green color one right here, which is present between xylem and phloem, which is present between xylem and phloem, is nothing but your fascicular vascular cambium. It is your fascicular vascular cambium. Fascicular vascular cambium. Okay, the green color one. Now what about the red color one? The red color one is your interfascicular cambium. It's going to be interfascicular cambium. And this interfascicular cambium, which can you see this here? Interfascicular cambium, which is present there, it's a type of secondary meristem. This one right here is a type of secondary meristem, and this is a primary meristem. This one is a primary meristem. Now this interfascicular cambium, is a result of dedifferentiation of medullary rays. Initially in this region right here, we had medullary rays initially. Medullary rays. Now those medullary rays underwent dedifferentiation. They underwent dedifferentiation and we ended up getting the interfascicular cambium. And this interfascicular cambium is nothing but secondary in origin. It is secondary in origin. This interfascicular cambium is secondary in origin. The red color one. Green color one is fascicular vascular cambium, which is primary in origin. Did all of you understand the difference now? Do all of you understand the difference between interfascicular cambium as well as your intrafascicular cambium? This is intra, this is inter. Intra is between xylem and phloem. Interfascicular cambium is between the vascular bundles. So this is one vascular bundle, this is second vascular bundle. Between the two vascular bundles, with the red color one is, between the two vascular bundles we have the interfascicular cambium. Where do we have intrafascicular cambium? Intra or fascicular vascular cambium is present, this green color one, between xylem and phloem. Between xylem and phloem we have the fascicular vascular cambium. Did all of you understand the difference now? Did all of you understand the difference? Yes or no in the chat quickly? Understood? Yes. Now let's see the next part. Following the divisions of the cell in both primary as well as your secondary meristem, the newly formed cells become structurally and functionally specialized and lose the ability to divide, that is dedifferentiation. Such cells are called as permanent mature cells. So when the plants undergo dedifferentiation, we get permanent cells. Okay, permanent cells. This is differentiation. Just simple differentiation. Simple difference, not dedifferentiation. When the plants undergo dedifferentiation, we get meristematic again. Differentiation constitute the permanent tissue. During the formation of primary plant body, specific regions of the apical meristem produce dermal tissues, ground tissue and vascular tissue. Basically, they are telling you the growth of the plant is happening because of primary meristem. That's all. That's all is the entire thing. Now, see here the examples of lateral meristem or lateral meristem is intrafascicular cambium, interfascicular cambium, as well as cork cambium. Three different examples for cambium here. Intrafascicular cambium, interfascicular and cork cambium. Intra is also called as fascicular vascular cambium. Remember that point. This is primary in origin. This is secondary in origin. This is also secondary in origin. Also secondary in origin. Done. Done, done, done. Look at this question. Look at this question. Axillary buds are derived from the activity of. How do you get axillary bud? That is, when the shoot apical meristem is growing, some cells are left behind. And those some cells that are left behind in the leaf axis is called as axillary bud. So what is the answer? The answer is going to be apical meristem. So as the apical meristem is growing, some cells are left behind. Next question. Consider the following plant tissues. NEET 2023 question. Consider the following plant tissues: axillary buds, fascicular vascular cambium, interfascicular cambium, cork cambium, and intercalary meristem. Identify the lateral meristem among the following. You are supposed to tell which one is a lateral meristem here. Which one is a lateral meristem? All of you know this one is, and this one is. All three are lateral meristem. So answer is B, C, D here. B, C, D. Option number 2. So students, let me tell you, anatomy is a chapter which requires a much more conceptual understanding. So if you understand the concept, all these questions becomes very easy for you. They become very easy for you. Okay. Look at this question now. Interfascicular cambium is present between. Where do you find inter, not intra, not intra, interfascicular cambium? Interfascicular cambium is found between where? Between the two vascular bundles. If you go back to the diagram, students, here, where do you find the interfascicular cambium? Interfascicular cambium is found between two vascular bundles. Between the two vascular bundles, we find the interfascicular cambium. Right? Two vascular bundles. Between the two vascular bundles. Done. Such an easy thing. Such an easy thing. Now, students, with that, your meristem tissues is over. I know I'm deliberately going very slow because if I rush anywhere, right, the concept will not be in your mind and you'll end up by hearting all this. So I don't want you to by heart, I want you to understand the depths of it. So I'm going very slow, it's fine because we have time right now. So I'm going to go slow, understand every concept one by one. Okay. Now let's understand something called as permanent tissues. Now I've told you there are two types of permanent tissues, haven't I? There are two types of permanent tissue. We have something called as simple permanent. We have something called as simple permanent as well as complex permanent. Simple permanent tissue and complex permanent tissue. What is the difference? What is the difference between simple permanent and complex permanent? In the case of simple permanent, it is made up of only one type of cell. It is only made up of only one type of cell. While your complex permanent tissue, students, complex permanent tissue is made up of more than one cell. It is made up of more than one type of cell. More than one type of cell. For example, xylem and phloem. Yes, xylem and phloem here. Xylem and phloem. Xylem and phloem. Here we have parenchyma, collenchyma, and sclerenchyma. Yes, parenchyma, collenchyma, and sclerenchyma. Done. That's the difference between simple and complex permanent tissue. Okay. Now let's understand simple permanent tissue today. Today class is going to be ending at simple and complex permanent tissues. That's where we are ending the class today. Okay. Because before we go to the sectioning, before we understand monocot, seed, dicot, before we understand the root of monocot and dicot, stem of monocot and dicot, as well as root of leaf of monocot and dicot, you need to understand all of this xylem and phloem. You need to understand what is parenchyma, collenchyma, sclerenchyma if you want to understand the sectioning. Okay. So don't worry, sectioning also I'll be teaching you by writing every single points. Notes will be made for you every single step of the way. Okay. Now let's understand what is parenchyma, collenchyma, and sclerenchyma. Now, first of all, can we start with parenchyma now? Let's start with parenchyma. So before we start the parenchyma, we'll have a 5 minutes break. I'll go drink some water and come. I forgot to get my water here. So let's start the parenchyma exactly at what's the time right now? The time right now here is 3:20. Okay. Exactly at 3:25 we'll start. Exactly at 3 3:25 p.m. we'll resume the class. 3:25 p.m. we'll resume the class. Okay? Done? 3:25 we'll resume the class. So all of you can also take a breather, drink some water and chill for a while. Okay? 3:25 we'll start the class again. 5 minutes, I'll drink some water and come. Students, can we start now? Can we start with the parenchyma now? Yes, come back soon. I feel everyone ran away now. Did everyone run away? Yes. Nice. Yes, everyone is back. Now, now let's start with the. Let me keep the pen. Yes, here. Students, tell me till now, is the chapter been difficult? Yes or no? Is the chapter is easy or difficult? Anatomy. Yes, I know. Meristem. You just have to read the chapter once, watch the session one more time, and you will understand it in a much more better manner. Just go through it one more time. You will have ultimate confidence over it. So other than that, the chapter is very easy, right? Now, when we go to the different types of tissues, or when we learn the different sectionings, it could be stem, root, or leaf. Similar fashion, I'll make you notes. Write down those notes, practice those notes, and you're good to go. And where do you find all of these notes? All of these notes you will be finding on my Telegram channel. On the Telegram channel, you will find all of these notes. All of these notes will be given to you on the Telegram channel. Baswat Sir Biology, you can find all the Telegram channel. Right? Now, with that being said, let's understand parenchyma. Now, when we hear parenchyma, what comes to your mind? Living or dead cell? Quickly in the chat. I want everyone to be active in the chat now. Everyone to be active in the chat. I will ask you a lot of questions. I want quick answers. Is the parenchyma a living tissue or a dead tissue? Quickly. Is parenchyma a living tissue or a dead tissue? Obviously, students, a parenchyma is a living tissue. It is a living tissue. Now, is it a common tissue or is it a rare tissue? Is it a common tissue or rare tissue? Obviously, it is a very common tissue. It is a very common tissue. It is a very common tissue. Now, where do you find it? Where do you find the parenchyma? The parenchyma can be mostly found in the majority of the organs. It can be found in the majority of organs. Major organ of the plants. It could be a root, it could be stem, or it could be leaf. It could be root, it could be stem, or it could be leaf. You find everywhere. You find it everywhere. Root, stem, and leaf. Everywhere we find the parenchyma. Where we find the parenchyma. Now, what is the shape of the cell here? The shape of the cell is going to be something called as isodiametric cells. What is the meaning of iso? The meaning of iso is same. The meaning of iso is same. Diametric means diameter. The same diameter is called as isodiametric. Can you see? Isodiametric. Same diameter. Isodiametric. Yes. The cell's shape is going to be isodiametric. Isodiametric. Apart from that, it can also be elongated in some cases. It could be spherical in some cases. Yes. It could be spherical, or it could be even oval in some cases. So the shape can vary. The shape can generally vary when it comes to parenchyma. Now, what about the cells? The cells here are going to be thin cells. Thin cell wall. Very thin cell wall here. Very thin cell wall. Now, what about the intercellular space here? Quickly in the chat. Do you find intercellular space here or not? If you look at this diagram right here, can you see some little intercellular space? Very less or almost absent in some cases. Yes, intercellular space can be very less. Intercellular space is very less, or it is completely absent. Intercellular space is less, or it is going to be completely absent. Intercellular space. Done. Now, what about the functions? What about the functions now? What about the functions? Functions of parenchyma. Functions of parenchyma. Now, the main function of parenchyma is obviously storage. It is going to be obviously storage. Everyone type storage in the chat. So once I mention any point, I want everyone to type in the chat the point so you'll remember it better. Or if you're writing it, it's even better. The main function of your parenchyma is storage. It's going to be storage. Now what if a parenchyma cells? What if a parenchyma cell has chlorophyll? Can it do photosynthesis? Yes, it can do photosynthesis. Yes, it can do photosynthesis. Done. And we call a parenchyma cell with chlorophyll, we call it chlorenchyma. We call it chlorenchyma. Yes, chlorenchyma. Done. Now what if it has certain gas chambers? Yes, it has gas chamber. We call that aerenchyma. We call that aerenchyma. Yes, aerenchyma. Done. If it has gas chamber. Now what is the other function? It can also help in buoyancy. Yes, it can also help in buoyancy. That exchange is written here anyway. Now some of the functions of parenchyma, some parenchyma can become secretory also. It can become secretory. That is, it can start producing certain type of secretions in the case of parenchyma. In the case of parenchyma. So if you look at the entire points of parenchyma, it is going to be living tissue, common tissue, isodiametric, elongated, spherical, or even oval in some cases, the shape, thin cell wall, interstellar space is absent or it is very less. Function is storage, photosynthesis, gas chamber, and secretory in some cases. It can also become secretory in some cases. That is the functions of parenchyma. Any doubt here? Quickly ask me. Any doubt here? Quickly ask me about parenchyma. Done. Now, what about collenchyma? What about collenchyma? First of all, tell me, is the collenchyma living or dead? Tell me, is the collenchyma living or dead? So, similar to parenchyma, collenchyma is also a living. It is also a living mechanical tissue. It is a living mechanical tissue. Living mechanical tissue. Yes. Now, in the case of parenchyma, we notice some form of interstellar space. But in the case of collenchyma, the interstellar space is completely absent here. It's going to be completely absent here. Interstellar space here. Now, what about cell wall here? In the case of your collenchyma, the cell wall has pectin deposition. Pectin deposits. Pectin deposits can be found here. Pectin deposits can be found here in the corners. Can you see? In the corners, we can see the pectin depositions in the case of collenchyma. Pectin depositions. Done. Now again, the shape again will vary here. The shape can vary from oval, polygonal, and different types of shapes are there, similar to your parenchyma. Different types of shapes are there. Okay. Now remember this extra point. Collenchyma can also contain chlorophyll. It can also contain chlorophyll. Also can contain chlorophyll. Now tell me the functions here again. Then what could be the functions here? Functions of collenchyma. Obviously the main function is going to be mechanical support. It's going to be mechanical support. The main function of collenchyma. Apart from that, it can also do photosynthesis. Collenchyma can also do photosynthesis. Why? Because of chlorophyll. Because of chlorophyll. Yes. Done. And where do you find collenchyma? I forgot to ask you. Where do you find collenchyma? Location of collenchyma. Tell me, where do you find collenchyma? Students, this is the most important point. Everyone should remember this. One of the most important points. Where do you find collenchyma? Collenchyma is only found in the, first of all, it is only found in dicots. It is only found in the case of dicots, and that too only in the case of, mostly in the case of stem. Most in the case of stem. In the leaf also it is present, but only in the case of petiole. Only in the case of petiole. It is completely absent. It is completely absent in the case of monocots and roots, as well as roots. It is completely absent. That is 𝐂𝐤. Is this point clear to all of you? Absent in the case of monocot and roots. In dicot stem and petiole it is present. Now, have you seen the young stem? The young stem is little flexible. Yes. And the leaf petiole is also very flexible. Elasticity is there. Yes. That elasticity or flexibility of petiole as well as the young stem is because, for the only reason, is 𝐂𝐤. Is 𝐂𝐤. So it gives elasticity to the plant. Provides elasticity to the plants. Done. Elasticity to the plant. That is, petiole can be bent and stem can also be bent. That is because of 𝐂𝐤. Now, what about 𝐶𝐤? Tell me, is it dead or living? First of all, is the 𝐶𝐤 dead or is it living? Is it dead or is it living? 𝐂𝐤. So, 𝐶𝐤. Obviously, students, 𝐶𝐤 is simple dead tissue. It is a simple dead tissue. Simple dead tissue. That is your 𝐶𝐤. Simple dead tissue. That is your 𝐂𝐤. Now, what will be the main function here? The main function of 𝐶𝐤 is obviously mechanical support. It's going to be mechanical support here. Mechanical support. When it comes to 𝐶𝐤, mechanical support. It has long, narrow cells. Long, narrow cells. All of the cells will look like this. Long, narrow cells. Long, narrow cells with lignified cell wall. Lignified cell wall. Remember, in the case of 𝐂𝐤, we had pectin depositions. Here we have lignin depositions. Lignin depositions in the case of 𝐶𝐤, pectin depositions in the case of 𝐂𝐤. That is the main difference. And it also has pits. What are these pits? I'll tell you in the next few minutes. See here. And it can be divided into two categories. 𝐶𝐤 can be divided into fibers and sclerets. Fibers and sclerets, what is the difference? Listen very carefully. In the case of fibers, we have long, narrow. Can you see? Thick-walled, elongated cells, first of all. They are thick-walled, elongated cells. They occur in groups. Cell wall has pits. What is this pit? Understand very carefully, students. What is pit? What is pit? Okay. Now, the outer part is the cell wall. The inner part is the plasma membrane. Right? Inner part is cell membrane. Inner part is plasma membrane. Okay. Now, between them there is lignin depositions. There is lignin depositions in the cell wall. Lignin depositions. We have lignin deposition is here. Yes, we have lignin deposition here. Complete lignified cell wall. Now, in this lignified cell wall, there are certain gaps. Can you see? There are certain gaps here. These certain gaps in the lignified cell wall. These gaps in the entire lignified cell wall is called as pits. They are called as pits. So, what are pits, students? Pits are nothing but the gaps in the lignified cell wall. In the lignified cell wall, there are gaps. The gaps are called as pits. Done. Is that point clear to all of you? If you're able to understand this concept, quickly smash that like button, all of you now. Quickly smash that like button if you're able to understand this concept of pits. Right? They have simple and border pits. simple pits and bordered pits. That is fibers. Now what about scleroids? Compared to fibers, they are more thickened, their shape can vary, and they have a very something called as narrow cavity or narrow lumen. Now if you open your NCERT, you can see a line. If you open a NCERT, you can see a line saying that the lumen is completely obliterated. The lumen is said to be completely obliterated. The meaning of obliterated is that the lumen is completely filled and consumed by the lignin depositions. The complete lumen is completely filled with lignin depositions. That is called as obliterated lumen. Done. And where do you find it? That is a PYQ. That is a PYQ right here. It is found in the fruit walls of nuts, fruit pulp of guava and pear, seed coats of legumes and tea leaves. This right here is a PYQ, previous year question. That is, where do you find the entire sclerets? Where do you find the entire sclerets? That is a PYQ. That is a PYQ right here. Where do you find sclerets and fibers? Done. See the question here. Which of the following is not a character of 𝐂𝐤 𝐐𝐤? Not a character of 𝐂𝐤 𝐐𝐤. You can read the entire thing. They provide mechanical support to the growing parts. Yes, they do. They occur in the layer below the epidermis. Yes, I told you here. Here we learned in 𝐂𝐤 𝐐𝐤, I think I didn't mention. I told you it is found in the case of stem. It is found in the case of stem. So in the case of stem, the outer layer is called as epidermis. The outer layer is called as epidermis. Just below the epidermis, we have the hypodermis. Hypodermis. And this hypodermis right here, it is made up of 𝐂𝐤 𝐐𝐤. Hypodermis is made up of 𝐂𝐤 𝐐𝐤. Yes. They consist of cells with thick corners due to cellulose depositions. Cellulose and pectin both. Cellulose and pectin both are there. Both are there. Right? Done. The last thing is, they are usually dead and without protoplast. No, this is a wrong statement, because we know 𝐂𝐤 𝐐𝐤 is a living tissue. 𝐂𝐤 𝐐𝐤 is a living tissue. Done. Next question. A type of tissue commonly found in the fruit walls of nuts. Now students, let me tell you, here you need to be very specific. If you tell 𝐤𝐤 𝐐𝐤, you will not get a marks. You need to tell scleroids. You need to tell sclerets. That is the actual answer. You can't tell entire. You can't just tell that 𝐤𝐤 𝐐𝐤. No. You have to be very specific here. Now with that being said, your entire simple permanent is over. Entire simple permanent is done and dusted. Okay. Now we go to complex tissue. Complex tissue, we will mainly study about the xylem and phloem. The first thing you need to understand here is they are not dividing cells. They are not dividing cells at all. They are very simple cells. Okay. They are not dividing cells at all. Okay. They are not dividing cells at all. They are the non-dividing cells. Okay. Xylem and phloem. Now here I told you the main definition of complex cells is what? They are made up of more than one type of cell. They are made up of more than one type of cell, right? More than one type of cell. That's the definition of complex tissue. Why is it? Yes. Yeah.
Hey guys.
I'm full no Josh only.
No Josh, all are sleeping, huh?
All are very sleeping. From DNA some are coming now.
Yeah. See, Kanmani, she knows she won't come.
Yeah.
Just came from class. Oh, DNA.
Yeah. You only teaching, no?
Yeah. I'm just reading who's
Oh, you're reading.
Yeah. Block which guy?
Done.
Done, okay. Hi guys, how is the Josh, yeah?
Yeah. There is no Josh, ma'am.
What is this? What to do, sir?
I don't know, ma'am. We should stop only, no, Haran.
We should stop everything.
We should not teach on YouTube only.
Yeah, we should only teach. Hello, we should teach what? Only on batch we'll teach, huh? Then you'll miss us on YouTube.
Yeah, then they'll tell, ma'am, you're not teaching on YouTube only, or you're concentrating on paid batches. Because when we do on YouTube, no one really shows up. So.
That's because it's free.
Yeah. Free things are not appreciated in this world, no, ma'am?
Oh God.
Yeah.
How much more, sir?
I don't know, ma'am. I think I'll be finished in half an hour, maximum.
Okay.
Don't, we'll eat later. I'll just go out. Okay. So that was Gopika ma'am. I think she has an eye infection, ma'am. Or ma'am has rubbed her eyes very vigorously and it has gone. But it's fine. See, this is what we happen. So even though we are tired, we are exhausted, we are not healthy, we still take classes here. Because we know the importance of every single day. Right? Every single day we know one day if you miss studying one day, you will actually be lagging behind. To make sure that you do not even miss a single day of studying here, whatever happens, we take a class. Right? Unless it's a very important business, we always end up taking class either on paid batch or on YouTube. If I'm not taking class here, I'll be taking on paid batch. If I'm not taking paid batch, I'll be taking here. The same goes for every single teacher on the channel. Right? Every single teacher on the channel, we keep doing this to make sure the entire flow of your education is not stopped. Right? Now, let's start with the complex tissue now. Let's start with the complex tissue. The first, if you look at the complex tissue, we have two types. First one is xylem, other one is phloem. Xylem and phloem. Now, under xylem, under phloem, we have different types of xylem, different type of phloem. All that we will do in today's class only. Everything will be covered in today's class. Okay. Now, but here we have something called as primary xylem and secondary xylem. Let me tell you, primary xylem is a primary tissue, primary permanent tissue, while your secondary xylem is secondary permanent tissue. Secondary xylem is a result of secondary growth. Result of secondary growth. The same goes for the secondary xylem, the secondary phloem. The secondary phloem is a result of secondary growth. Secondary growth. That is secondary xylem, secondary phloem. Okay. That point needs to be clear for all of you. That point needs to be clear for all of you. What is it? Protoxylem and metaxylem? Protoxylem is the first formed xylem. Metaxylem is formed later on. The same goes for protophlloem and metaphlloem. Protophlloem is first formed. Metaphlloem is formed later on. It is formed later on. That is the difference. Now let's understand one by one the xylem. Let's understand the xylem now. Okay. Now students, I want everyone to write down these points. It will be easier for you to remember also later on. What about xylem? First of all, tell me, xylem is a conducting tissue. Yes, it is a conducting tissue. It is a conducting tissue. Done. It is a conducting tissue. Now, xylem, is it living or non-living? Is the xylem living or non-living? It is actually both. It is living as well as non-living. Living as well as non-living. That I'll tell you in the next few minutes. How many of them are living, how many of them are dead, we'll discuss later on. Now, what is the main function of xylem? You've been learning from class 12th, class 10th. The main function of xylem is conduction. The main function of xylem is basically conduction of water. Main function is conduction of water and minerals. Water and minerals. Yes, that is the main function of xylem, conduction of water and minerals. Done. Now, tell me, is the flow of water unidirectional or bidirectional? For example, water is present here. With the help of xylem, all the water is moving upwards. All the water is moving upwards. It is only a unidirectional flow. From the top, water will never come down. From the top, water will never come down. It is always from the bottom to the top. That is called as unidirectional flow of water. It is always unidirectional flow. Unidirectional. It is always unidirectional. Done. Now, under xylem, we have tracheids, vessels, parenchyma and fibers. Now, quickly tell me in the chat, how many of them are living and how many of them are dead? Go. Time starts now. Go. How many of the following are living? How many of the following are dead? Your tracheids is non-living. Vessels is non-living. Parenchyma is living. Xylem fibers are again non-living. Again they are non-living. Understood? So non-living, non-living, living and non-living. That is the story of the xylem. Now let's understand tracheids now. One by one we'll go about each one of the following. Now what are tracheids? Tracheids are basically long tube-like structures. They're long tube-like structure which are unicellular. They are unicellular, long tube-like structures, and some of them can also have something called as tapering ends. Can you see? Long tube-like structures with a tapering end. Elongated cells, tapering ends, thick lignified cell wall. Why? Because they are dead. They are dead cells. So if they are dead cells, obviously they'll have very high thickened cell walls. The reason they're dead cell is because they have high lignified cell wall. If the lignin deposition is there, do you think any of the following material can enter inside? No. Nothing can enter inside. That's why it's a dead cell. That is why it is a completely dead cell. So highly lignified cell wall here. Highly lignified cell wall. Inner layer of the cell wall have thickening that vary in the form. Dead cells without protoplast. One of the main water transporting elements of xylem. It is one of the main reasons where how the water starts conduction. Conduction of water happens. That is because of tracheids. Now what about vessels? Vessels are on the other hand multicellular. They are multicellular in nature. Multicellular. If you can look at the structure of your vessels, this is one vessel, this is another vessel, and they are called as vessel members. They are called as vessel members. So on one top of another, we have one vessel, on top of we have one more vessel. Vessel. So all the different types of vessel members will be arranged one after one another, like this. One vessel, second vessel, third vessel, fourth vessel. And between the vessels, students, between the vessels, we have some form of perforations. We have something called as perforations. Why perforations? The main reason for this perforation is that so that water can easily flow. Water can easily flow. If there is no perforation, then water won't be able to flow. Water won't be able to flow. So we have something called as perforations, also called as cell plate formation. Here we have the cell plate formation. Perforation, because of perforation, you see small holes here. Because of perforations, all the water can easily flow. All the water can easily flow. Long cylindrical tubes made up of many cells and called as vessel members. Each cell has lignified wall and large central cavity. Obviously, lignified wall here completely and large. You can see this middle part, central cavity is there. No protoplasm, that's why it's a dead cell. Interconnected through the perforations in their common walls. There are perforations and that's how they are connected to one another. Okay. A typical feature in angiosperm. Now that is very important because these vessels are not found in the case of gymnosperms. They are not found in the case of gymnosperms. Okay. It's only found in the case of angiosperms. Done. See the simple difference. Present in all vessel plants. They are present in only angiosperms. Narrow lumen, wider lumen. Highly thickened, less thickened than the tracheids. Do not have end plates. They have the perforated end plates. That's how one after another they are connected because of perforated end plates in the case of vessels. What is parenchyma? See, all of you know parenchyma, the main function is storage. So here also the main function will be storage only. Living cells, parenchyma, thin cell wall. Remember all the different types of properties of parenchyma. Living cells. Cell wall is made of cellulose. Function is what? Storage of food. Main function is storage of food here. Food. In the form of starch or fat. Okay. And even substances like tannin. That is xylem parenchyma. Now again, xylem fibers will be dead. The dead tissues. Highly thickened walls. Now here, usually the lumen is absent here. Can you see any lumen here in the middle? You cannot see any lumen here. The entire structure of the xylem fibers is going to be completely, the lumen will be completely obliterated. The lumen of xylem fibers is completely obliterated, means completely filled by lignin depositions. It is completely filled by lignin depositions. But sometimes what happens is, it can have some form of septa. Can you see? Some of them can have small septa, that is demarcation. The septa can be absent in some cases. So, septate or aseptate. Done. Now students, I want to understand a few more concepts here. We have the primary xylem and secondary xylem. Primary xylem and secondary xylem. And we have something called as protoxylem and metaxylem under primary. Under primary, not under secondary xylem. Under primary xylem, we have protoxylem and metaxylem. Now, based on the arrangement of protoxylem and metaxylem, we have two conditions called as 𝐂𝐴𝐡𝐞𝐜 condition and NEET condition. What is the difference? Listen to me very carefully. We have something called as NEET condition and 𝐂𝐴𝐡𝐞 condition. Now, what exactly happens in the NEET condition? See, if this is the pith, this is the pith. If protoxylem is towards the pith, you see here is the pith, here we have protoxylem. If the protoxylem is towards the pith, then it is called as NEET. Simple. Protoxylem towards the center, that is towards the pith, and metaxylem towards the periphery, that is called as NEET. Now, what is 𝐂𝐴𝐡𝐞? X means outside, X means periphery. So, when the protoxylem is towards the outside, when the protoxylem is away, 𝐂𝐴𝐡𝐞, X outside. If protoxylem is towards outside, it is called as 𝐂𝐴𝐡𝐞 condition. It's called as 𝐂𝐴𝐡𝐞 condition. That is protoxylem on the periphery. Protoxylem on the periphery, that is towards the outside, is called as 𝐂𝐴𝐡𝐞 condition. And 𝐂𝐴𝐡𝐞 condition is mainly found in the case of roots, and NEET condition is mainly found in the case of stem. NEET, stem, 𝐂𝐴𝐡𝐞 in the case of roots. 𝐂𝐴𝐡𝐞 in the case of roots. Is that point clear to all of you? Based on the position of metaxylem and protoxylem, we can tell 𝐂𝐴𝐡𝐞 and NEET. What is NEET? Protoxylem towards the pith, NEET. Protoxylem away from the pith is going to be 𝐂𝐴𝐡𝐞. 𝐂𝐴𝐡𝐞 is away. That's all. That's all is 𝐂𝐴𝐡𝐞 and NEET concept. Look at this question. And students, remember, both of them are part of primary xylem, not secondary xylem. Only primary xylem. This question you see, 2023. NEET and 𝐂𝐴𝐡𝐞 term often used to describe the position of secondary xylem in the plant body. 𝐂𝐴𝐡𝐞 condition is most common feature in the case of roots. Yes, it is found in the case of roots, 𝐂𝐴𝐡𝐞. But just now I told you, it is not secondary xylem, it is going to be primary xylem. So can I tell the first statement is wrong, second statement is correct? Yes. 1 is the answer. Okay. Now let's understand phloem now. That was your xylem. That was the xylem. Let's understand phloem now. Let's understand phloem. Obviously, phloem is also a conducting tissue. It is also a conducting tissue. Yes, phloem is a conducting tissue. What are the main function of phloem? Transport of food. Transport of food. Main function. Yes. Now, where is food produced? The food is produced in the leaf generally. From the leaf, will the food go to the other parts of the plant? For example, there is a flower on top. The food which is prepared in the leaf, will the food go to flower? Yes. The food which is produced in the leaf, will it go to the roots? Yes. So can I tell the transport of food with the help of phloem is bidirectional? It is bidirectional. It is bidirectional. Transport of food is bidirectional here. Okay. Now, apart from that, I want you to understand there are different types of phloem also. For example, we have sieve tubes, companion cells. Together they are called as sister elements. We have phloem parenchyma and phloem fibers. How many of them are living and how many of them are non-living again? That is simple. It's the complete opposite of xylem. Here, your sieve tubes are living, companion cells are living, phloem parenchyma is living. The only non-living part of phloem is going to be phloem fibers. The phloem fibers are going to be non-living. The only non-living part is going to be phloem fibers, non-living. Rest are living. Let's understand sieve tubes now. Let's understand sieve tubes and companion cells. I have mentioned them together with each other so you can understand better. Okay, they're called as sister elements. Okay, first of all, CU tubes are long tube-like structures. Can you see here? They are long tube-like structures. Here. Long tube-like structures. That is CU tubes. Arranged longitudinally. They're arranged longitudinally. Yes. End walls are perforated in the sieve-like manner, similar to your vesicle vessels. End walls are perforated. Can you see? End walls are perforated here. End walls are perforated. So transportation can happen. Yes. End walls are here perforated. Here also. Perforations are here. Yes. Perforations are there. One minute for this. This is your entire CU tube. I don't think you're able to see it. The red color one is the CU tube. Can you see now, red color one? This right one is the CU tube elements. Now can you see this the tapering end one? This tapering end one, there's green color one, tapering end one. I'm drawing it with pink. This tapering end one is called as companion cell. They are the companion cell. The red one right here is going to be CU tube and pink color one is companion cell. Okay. Now here end walls are perforated. You see, end walls are perforated here. End walls are perforated. Mature sieve elements have peripheral cytoplasm and large vacuole lack nucleus. Your complete CU tubes will completely lack nucleus, and the entire function of CU tube is going to be controlled by companion cells because they don't have nucleus on their own. So if you look at the function is controlled by the nucleus of companion cell. Specialized parenchyma cells closely associated with the CU tubes. Close association. CU tubes and companion cells are connected by pit field present between the common longitudinal walls. Here there are pit fields. Here. Let me draw it for you, you will understand better. Students, let me draw it for you. This is a companion cell. This is a companion cell. Next to the companion cell, we have the CU tubes. This is how it looks like. Here there is nucleus in companion cell. This is companion cell. This is going to be companion cell. This one is CU tube. CU tube. Okay. The CU tube right here, I told you, long tube-like structure. Here. Arranged longitudinally. Yes, longitudinally. End walls are perforated. Can you see? The end walls are perforated. Yes. Mature CU elements has peripheral cytoplasm. Here, cytoplasm is here. Okay. And large vacuole. There will be a large vacuole here. And it will have no nucleus. It will have no nucleus. While this is parenchyma cells, companion cells. They are in close association with the CU tube elements. But how are they connected, students? How are they connected? Here between them you can see some pit fields. You can see some pit fields here. That is connection here. Pit fields are present between companion cell as well as CU tube cells. There is some form of pitted fields are there. That is, and they help in what? What is the main function? This is the PYQ. Main function of companion cell is they maintain pressure gradient inside the CU tubes. Why do they maintain? Because they have nucleus. CU tubes do not have nucleus. So they help in maintaining. Done. Okay. Then we have phloem parenchyma. And students, remember, extra point I'll tell you, which is also given you in NCERT. your companion cells are present only in angiosperms. They are present only in angiosperms. They're only present in the case of angiosperms. In the case of gymnosperms, they have sieve elements. They have something called as albuminous cells. There's something called as sieve cells are present. Even sieve tube elements are also absent in the case of gymnosperms. There are sieve cells and albuminous cells. Okay, these two are absent in the case of gymnosperms. They are exclusive to angiosperms, higher plants. See, parenchyma, obviously main function is going to be, see, there are also long tapering cylindrical cells, dense cytoplasm, similar to in the case of your parenchyma cells. Absent in the case of monocots, very important point. Phloem parenchyma is absent in the case of monocots. Plasmodesmata connections are there. Main function is obviously parenchyma, storage of food material. Fibers, again dead cells first of all. Made up of sclerenchyma, dead cells. Absent in primary phloem. They are only present in secondary phloem, that is later formed in life. Elongated or unbranched, long tapering ends, pointed needle like apices. Cell wall is very thick. That they are dead cells. This is your phloem fiber which they use to make coir and everything. Okay. This is your entire phloem. Protophlloem, metaphlloem. Friends, with that we will stop today's class. In the next class we will start with tissue system. So before we go, let me know in the comment section how was the class, and here is your homework question for today. Here is the homework question. Easy homework question, you can tell. If phloem in gymnosperm lacks, what are the missing things in gymnosperms? Okay, this is your homework question. This is your homework question. Okay, this is a very chill class because this is a very easy chapter. I want to teach in a much more slow pace, so all of you will be able to appreciate and understand every single point given here. Okay, so with that being said, I will take off right now. Please smash that like button, all of you, and let me know in the comment section how was the class, and any difficulties you faced in any of the following topics. Let me know in the comment section, and I will see you in the next class, which is actually tomorrow. Next class is tomorrow 6 p.m. Tomorrow 6 p.m. I will be taking the class. Okay, so with that being said, take care all of you, and bye bye.